Hello everyone, I'm Accadius Lunayo from the Center for Biotechnology and Bioinformatics at the University of Nairobi. Today I present to you a project that was developed at the center in a bid to identify and develop therapeutics against a zoonotic parasite, Cryptosporidium parvum, that predominantly causes diarrhea in children and neonatal livestock. As we all know, diarrhea isn't pleasant, one of the pleasant diseases that you can have. It's actually, apart from that, very deadly. In developing countries and generally worldwide, diarrhea is the second leading cause of death by infection in children who are under 5 years. And it causes more than 499,000 deaths per year. A particularly large share of this particular disease burden has been attributed to the parasite Cryptosporidium. As you can see in the diagram, the parasite predominantly attacks the intestines and causes severe diarrhea. Of all the parasites in the genus Cryptosporidium, Cryptosporidium parvum is the major zoonotic species that causes diarrhea predominantly in both children and newborn livestock. And these diarrheal syndrome cause death and retarded growth to children, as well as due to malnutrition and newborn livestock, and it also causes significant morbidity and mortality in developing countries. In HIV patients, it causes severe cholera-like illness, which leads to wasting and malabsorption of ARV, and this results to reduced life expectancy and very high mortalities in individuals with immunocompromised immune systems. Cryptosporidium parvum is highly prevalent because it is resistant to almost virtually all disinfectants, including chlorination, and that makes it very difficult to eliminate from the environment. And despite all that, the treatment options are severely limited, and there is no vaccine that is available. Our study focused on specifically 3 aims, and these aims were to determine the 3D structure of the C. parvum 18S ribosomal RNA using computational modeling approaches, and specifically homology and de novo modeling, to identify the important motifs of this particular 18S ribosomal RNA 3D structure, and finally to explore the potential of these motifs and drug targets through structure-based molecular docking. As part of methodology, we employed and utilized in silico sequence-based methods to design and select drug compounds against the C. parvum. The workflow was very particular. It used a 3-step workflow as shown here. The first step was 3D modeling using de novo and homology modeling. The second step was identifying which particular regions of this particular ribosome could be targeted, what we'll call active sites, using blind docking. And the last stage was to identify how well these what we call docking points, generally referred to as pockets, bind to different drugs or compounds from different databases by redocking them and then carrying out molecular drug ability analysis and then molecular dynamics. Our results were pretty outstanding. Looking at the structure we had, we leveraged on the 3D cryo-EM ribosome structure of a closely related parasite, that is Toxoplasma gondii, and we managed to model a 3D structure of C. parvum with a root mean square deviation of 0.845 angstroms. Additionally, we used the information from the RNA structure and the movement to identify and dock a set of promising anti-infective-like compounds that can biologically target the function regions of the ribosome in C. parvum. As shown in the diagrams here, this was developed. The results indicated that there is a high sequence conservation between the 2 structures. We developed a secondary structure with a few points that were significantly different, the variable regions, and the difference between the model structure and the experimental structure was almost indistinguishable, as you can see from the T. gondii here and C. parvum. We got some very interesting results, and one of them was as you can see, this is the path for the mRNA in the ribosome, and this is a point where we got docked. Some molecules got docked, and we have been analyzing these molecules case by case to actually see how they can actually be used or explored further as drug targets. As I wind up my presentation, we actually know that Cryptosporidium presents many technical obstacles to efforts aimed at developing fully effective drugs against infection. And one of the most important one, and the most big roadblock, is the biology of the Cryptosporidium is poorly understood. And therefore, there is limited genetic tools for functional integration, interrogation and validation of potential molecular targets in the parasite. And here we are presenting and utilizing a set of in silico-based methods that we have developed atomic level 3D model of the 40S, and we're only using the sequence information, and we intend to use this to accelerate drug discovery process in cryptosporidiosis. And together, our findings have unveiled promising drug candidates that can be explored further for development of the much needed novel therapeutics against C. palmum infections. Finally, our approach can greatly advance the development of narrow spectrum anti-infectives against cryptosporidiosis. Having said that, I wish to acknowledge the University of Nairobi. This part of this work was partly funded by the University of Nairobi and partly funded by the National Research Fund. And finally, acknowledge the Ohio State University with specific mention of the International Affairs Office. Thank you very much.